Imagine losing 20 kilograms on semaglutide, tirzepatide, or another GLP-1 medicine. Your clothes fit again, your waist is smaller, and the number on the scale looks incredible. But then you look more closely. Your cheeks appear hollow, your skin looks looser, and weights that once felt easy now feel unusually heavy. Did the medication help you lose fat, or did it also cost you valuable muscle? And can two peptides that are spreading rapidly through the biohacking world, Follistatin and GHK-Cu, actually prevent the damage? Or are people using experimental biology to solve a problem that those peptides have never been proven to treat? Today, we follow the human evidence. I'm Sharif El-Jendi, and welcome to Peptide IQ, where we investigate peptide and GLP-1 claims using human clinical evidence, regulatory facts, and product quality science. In this investigation, we will answer 4 questions. How much lean mass do people actually lose? What really causes the appearance called Ozempic face? Could follistatin preserve muscle? And could GHK-Cu rebuild the face? I am a researcher and educator, not a medical doctor. This video is educational and is not personalized medical advice. Follistatin and injectable GHK-Cu are not established treatments for GLP-1 related muscle or facial volume loss. Never use an experimental substance based only on a video, online protocol, or personal testimonial. Now, let's begin with what the Dexa scans actually found. In the Step 1 body composition sub-study, researchers used Dexa scans to examine a smaller group of participants receiving semaglutide. After 68 weeks, body weight had fallen by approximately 15%. Total fat mass fell by more than 19%. Visceral fat, the deeper fat surrounding the abdominal organs, fell by more than 27%. But total lean body mass also fell by approximately 9.7%. Now look at Tirzepatide. In the SURMOUNT-1 Dexa sub-study, around 75% of the weight lost was classified as fat mass. The remaining 25% was classified as lean mass. So the concern is real. When people achieve substantial weight loss, they do not normally lose fat and nothing else. Some non-fat tissue is usually lost too. But this is where many viral videos make a critical mistake. Lean mass does not automatically mean skeletal muscle, and it certainly does not prove that GLP-1 medicines are stealing muscle directly. The difference matters enormously. Dexa places most of the body into 2 major compartments, fat mass and lean mass, but the lean compartment contains more than muscle. It includes body water, organs, connective tissue, and other non-fat components. That means a decline in Dexa lean mass cannot tell us precisely how much contractile skeletal muscle was lost, and it cannot tell us whether a person became weaker. This is why strength and physical function matter. A newer prospective semaglutide study found that lean mass fell during the early months of treatment and then stabilized. At 12 months, hand grip strength had actually improved. Other imaging research has found reductions in muscle volume, but also improvements in fat infiltration within muscle. So the scientifically accurate conclusion is not GLP-1 drugs destroy your muscles. The accurate conclusion is significant weight loss can include a meaningful reduction in lean tissue, and the risk may matter more for older adults, people with low starting muscle mass, severe calorie restriction, poor protein intake, or little resistance exercise. That brings us to the first proposed solution, follistatin. Follistatin is a naturally occurring protein involved in several signaling pathways. Its most famous target is myostatin. Myostatin helps limit skeletal muscle growth. That is why it is often described as a biological brake on muscle size. Follistatin can bind myostatin and reduce its activity. In animal models, increasing follistatin activity can produce substantial muscle growth. That creates a very attractive theory. If calorie restriction tells the body to reduce tissue, could blocking myostatin force the body to preserve muscle? Mechanistically, the idea is interesting, But follistatin is not a simple switch that controls only one pathway. It also interacts with activins and other biological systems involved in reproduction, inflammation, metabolism, and tissue regulation. A powerful mechanism does not automatically produce a safe or effective treatment, and the human evidence is very different from what peptide sellers normally describe. One of the best-known human studies involved only 6 people with Becker muscular dystrophy. Researchers did not give them an ordinary vial of follistatin 344. They used an experimental viral vector to deliver the follistatin gene directly into the quadriceps muscles. Some participants improved their distance in a 6-minute walking test. Others showed little or no improvement. The results were scientifically interesting, but this was a tiny, early-stage study in people with a serious genetic muscle disease. It was not a study of healthy bodybuilders, people using Semaglutide or Tirzepatide, or people injecting retail follistatin bought online. There is another important difference. The study delivered a gene designed to produce a specific follistatin form within muscle that is biologically and pharmacologically different from injecting an unapproved peptide product from a vial. The evidence does not show that follistatin guarantees muscle preservation during GLP-1 weight loss. It does not show that all weight lost becomes pure fat, and it does not establish that muscle gained through follistatin is automatically permanent. You may also hear that follistatin is safe when limited to a 20 or 30-day cycle, or that other research peptides must be combined with it to protect the tendons. Those are not validated clinical rules. The Peptide IQ verdict on follistatin is compelling mechanism, preliminary human gene therapy research, but currently unsupported as a treatment for preventing GLP-1-related muscle loss. Fortunately, experimental follistatin is not the only possible response to lean mass concerns. The strongest current clinical guidance begins with resistance exercise. Muscle needs a reason to remain. Mechanical tension from properly designed strength training provides that signal. Adequate protein also matters, but the right amount depends on body size, age, medical history, kidney function, total food intake, and training. That target should be individualized with a qualified clinician or dietitian. If nausea or early fullness makes eating difficult, the answer is not automatically to force down a large steak or copy an influencer's protein target. A clinician can help adjust meal size, food texture, medication escalation, nutrient timing, and treatment tolerability. And do not measure success with weight alone. Track waist circumference, gym performance, walking capacity, grip strength, and everyday function. A Dexa scan may be useful in selected people, but even Dexa requires careful interpretation. No research peptide can replace the basic anabolic signal created by using your muscles. Now, let's examine the face. Ozempic face is not a formal medical diagnosis. It is a popular term describing facial changes noticed after substantial weight loss. The appearance can include hollowed cheeks, more prominent folds, reduced mid-face support, looser skin, and wrinkles that suddenly become more visible. But this is not necessarily evidence that semaglutide is directly destroying collagen. A major part of the change appears to be simple volume loss. One radiographic study estimated an average loss of around 7% of midfacial volume for every 10 kilograms of total weight lost, with much of the reduction occurring in superficial facial fat pads. The more weight someone loses, the faster it happens, and the less skin elasticity they began with, the more noticeable the change may become. Age, genetics, sun exposure, smoking history, hydration, and starting facial structure can also affect appearance. And this phenomenon is not exclusive to Ozempic. Facial deflation can occur after major weight loss from medication, dieting, illness, or bariatric surgery. That distinction is essential because improving the skin is not the same as replacing lost facial volume. This is where GHK enters the story. GHK is a naturally occurring 3-amino acid peptide that can bind copper. Laboratory and animal research suggests that it can influence fibroblasts, collagen, elastin, wound healing, and extracellular matrix remodeling. Small topical cosmetic studies have also reported possible improvements in skin thickness, elasticity, fine lines, and visible skin quality. But notice the words I used: possible improvements in skin quality. That is not the same as rebuilding a lost facial fat pad. If weight loss has removed volume from beneath the skin, stimulating collagen cannot automatically recreate that missing three-dimensional support. And the human evidence is not uniformly positive. One controlled study examining topical copper tripeptide after laser resurfacing found no significant improvement in wound healing compared with the control treatment. Most importantly, I could not verify a controlled human trial showing that injectable GHK-copper prevents or reverses facial volume loss caused by GLP-1 treatment. The mechanism is interesting, but the claim that it produces an internal facelift is far stronger than the clinical evidence. Injectable GHK-copper creates a separate safety question. The United States FDA notes that compounded injectable GHK-copper may present immunogenicity risks because peptides can aggregate or contain difficult-to-characterize impurities. Human safety data for injectable use remain limited. You may see online instructions claiming that GHK-copper should be mixed in the same syringe with BPC-157 to neutralize discomfort. There is no reliable clinical evidence establishing that this mixture is chemically compatible, sterile, stable, safe, or guaranteed to become painless. Combining unapproved substances does not automatically create synergy. It can create an unknown product that has never been formally tested. The Peptide IQ verdict is: topical GHK-copper is not a proven treatment for Ozempic face, and it cannot currently be presented as a clinically established cure. I also want to separate evidence from personal experience. I have personally tracked changes in my waist, belt size, strength, weight, and smart scale readings while researching these compounds. At times, my waist measurement clearly changed while the BIA scale appeared to tell a completely different story. That does not prove the scale was deliberately lying, and it does not prove that a particular peptide created new muscle. BIA estimates body composition indirectly through electrical impedance and prediction equations. Hydration, food intake, exercise, testing time, temperature, and the device's algorithm can alter the result. That is why one smart scale reading should never be used to prove that someone gained muscle, lost visceral fat, or experienced a specific drug effect. Use consistent conditions, watch trends, and combine measurements with waist circumference, strength, function, photographs, and clinically appropriate testing. My experience can generate a question. Only controlled research can test whether the explanation is true. So, do Follistatin and GHK-copper actually solve GLP-1 related muscle loss and Ozempic face? Here is the evidence-based conclusion. First, substantial GLP-1 associated weight loss can include lean tissue loss, but most of the weight lost in the major body composition studies was fat, and lean mass is not identical to skeletal muscle. Second, Ozempic face is primarily associated with facial volume loss after substantial weight reduction. It is not proof that the medication directly destroyed the skin. Third, Follistatin has a powerful biological mechanism, but it has not been clinically proven to preserve muscle in people using GLP-1 medicines. Fourth, GHK-copper may have interesting topical effects on skin quality, but injectable GHK-copper has not been shown to restore lost facial fat or reverse Ozempic face. The evidence-based foundation remains far less exciting, but far more defensible. Progressive resistance training, adequate and individualized nutrition, monitoring strength and function, avoiding unnecessarily severe calorie restriction, and working with qualified clinicians when side effects, rapid loss, weakness, or nutritional problems appear. In the next Peptide IQ investigation, we will expose another body composition problem: why an expensive smart scale can show that you gained fat overnight, and which measurements actually reveal whether a GLP-1 treatment is improving your body composition. If this investigation helped you separate evidence from hype, press like, subscribe to Peptide IQ, and share it with someone using a GLP-1 medicine. I'm Sharif El-Gendi. Stay curious, question the marketing, and always follow the evidence.